Bismillahirrahmanirrahim. Welcome back, guys. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we will start from 51 MCQs, and these MCQs are actually related to the chapter number 3, sterilization technique, culture media, etc. Okay, now we already talked 50 MCQs in the 2 video lecture previously. If you miss, so you can go back and watch it. There will be also a link in the description, you can also check out it. Okay, we also talked 2 other chapter MCQs related to the microbiology, so you can also watch it into my channel. Okay, now today in this video lecture we are going to start with 51 MCQs. So let's start. The 51 MCQ says that the bacterial culture prepared by the Pewer culture, what we call that. This one MCQ is again repeated, so we mostly use inoculum. That is the Pewer culture technique. Okay, now separation of a single colony is what? So separation of the single colony is again isolation. This one MCQ is again repeated. B is the correct option. Growth period of the culture is, so growth period of the culture is mostly incubation. B is the correct option. Incubation is used to. Now at a temperature 160 centigrade for 1 hour, complete sterilization is what? So mostly this one sterilization is take place in the hot air oven. B is the correct option. In autoclave, the principle involved is, so moist heat principle is mostly involved. We can also say that the stream under pressure is also involved. So the correct option is D. Both B and C is the correct option. MCQs 56 says that the spores of bacteria which can withstand the moist heat also. So for this the MCQs is the correct option is Bacillus styrothermophilus. C is the correct option. MCQs 57 says that factor on which disinfectivity of a disinfectant depends. So it mostly depend upon on the concentration, time. So D is the correct option, all of the above. Now MCQs number 58 says that aldehyde which are most powerful disinfectant. So the most powerful disinfectant aldehyde is acetaldehyde and glutamyl aldehyde. Formaldehyde and glutamyl aldehyde. So both A and C is the correct option. MCQs 59 says that acridine dyes are most effective against. It is mostly effective against gram-positive bacteria. A is the correct option. The sterilization agent, so the sterilizing agent is, so the sterilizing agent is obviously ethylene oxide. A is the correct option. We used this technique for the plastic to sterilize the plastic material. Then we have 61 MCQs, salt of heavy metals used as a disinfectant. What we call that? We mostly call that thiomersal, phenol, mercury. D is the correct option, all of these. 62 MCQ says that culture are prepared by penetrating the inoculation loop with suspension into the medium. They are what? They are mostly step culture. B is the correct option. MCQ 63 says that the principle of the streaking method involved, so it mostly involved dilution. D is the correct option. MCQs number 64 says that culture media for fungi are, so for fungi the culture media is actually streaking. No, for fungi, sorry, for fungi the culture medium is subrod dextrose agar. D is the correct option. Okay, all type of the media is actually for the culture, for the fungi. Now spores of actinomycetes are very sensitive, killed at a room temperature. So the best temperature is which one? So the best temperature is 65 to for 30 minutes. B is the correct option. 66 MCQ says that the term that is used for the bacteria which can withstand pasteurization but does not grow at higher temperatures. So for the 66 the MCQs the correct option is the thermodiuric. C is the correct option. MCQs number 67 says that a common laboratory method of cultivating anaerobic microorganism is the correct option is C, pyrogallic acid and over the cotton. C is the correct option. MCQs number 68 says that alkalophiles grow at a pH of, so it mostly grow at a pH of 7 to 12 centigrade. The microorganism grow at a high salinity, we call that halophiles. B is the correct option. Non-lactose fermenting colony seen on a Macon key medium, E. coli is the lactose fermented, Klebsiella is the lactose fermented, Salmonella typhimurium, we can say this is the A is the correct option. This is the non-lactose fermented bacteria. MCQs number 71 say Wilson and Bailey medium is used for the isolation of what? It is mostly used for the isolation of Salmonella typhosa. B is the correct option. Laboratory diagnostic of the enteric fever is based on, so it's mostly based on the Vidal test. C is the correct option. Shigella was first isolated by, so it was first isolated by Shiga. A is the correct option. Which of the following are producing salmonella, gas producing salmonella? So for gas producing salmonella, the correct option is B, Salmonella enteritidis. 75 MCQ says the Kaufman White scheme is used to detect what? It is mostly used to detect Salmonella species. A is the correct option. So that's the other 25 MCQs related to chapter number 3. That is the 75 MCQs. Inshallah, in the next video lecture we will start from the 76 and will end with 105 MCQs, which will be our last video related to the chapter number 3. I hope this video is helpful for you. Thank you so much for watching.